All right. Well, g'day everybody. My name's Peter, and I'm going to be giving the workshop on using the DelayedArray package to analyze large data sets. I've pre-recorded the workshop and we'll be just playing the pre-recording from YouTube. I've just tweeted out the links to the recording itself as well as the workshop material. Some of the text when played back via video, so the sort of screen sharing part of the presentation, due to internet connection issues and YouTube and compression and the like, may be a bit hard to read. So I suggest that you have the workflow vignette available so that you can read the code more clearly. And I encourage you to follow along using the RStudio servers, because I've built into the workshop a couple of opportunities to sort of have a go yourself. So the sort of prepared material goes for about 40 minutes, and then we'll come back, should have about 10 to 15 minutes for discussions and questions. So I'm going to share my screen now and start the video. G'day, my name's Peter Hickey, and I'll be teaching you the workshop on effectively using the DelayedArray framework to support the analysis of large data sets. Welcome. Before we begin today's workshop, we will download some data necessary for it from the Experiment Hub. To do this, we load the Experiment Hub package into our R session. Once the package is loaded, we construct an Experiment Hub by calling the Experiment Hub function. The dataset that we will be using in today's workshop is a large single-cell RNA-seq dataset produced by 10x Genomics. To find this dataset, we will query the Experiment Hub with the phrase 10x brain data. There are a number of resources available from Experiment Hub that match this query. The one that we will be using in today's workshop is this one highlighted here. To download this resource, we access it from the hub by typing in the name of the resource. This will download the resource from Experiment Hub. This is a rather large dataset stored in the HDF5 file format and may take a while to download. The advantage of using Experiment Hub to download this dataset is that once it's downloaded to your machine, subsequent calls to Experiment Hub will use the local version of the dataset. That's all for now. We'll let this dataset continue to download whilst we begin the introductory material for today's workshop. The DelayedArray framework is led by Hervé Pages, one of the Bioconductor core team members. The reason I'm here today presenting this workshop is that I have been an early adopter of the DelayedArray framework, using it to analyse large datasets at the cutting edge of high-throughput biology. So I see myself as a bit of a crash test dummy for the DelayedArray package. I'm also a developer of packages that use and extend the DelayedArray framework, in particular those for analysing DNA methylation data. At the heart of what we do in Bioconductor are many core data structures. One of these is the summarized experiment, used to store array-like data and associated metadata. At its heart, a summarized experiment is an array of values with metadata on the rows, which are the features, and metadata on the columns, which are our samples. We may have multiple assays, so multiple arrays of data, and we may have some metadata on the experiment as a whole. This is all encapsulated in the summarized experiment object. Today's workshop focuses on the assay data, the actual measurements that are the basis for our analysis. Typically, these are stored as an ordinary R array or matrix. Ordinary R arrays are structured. For Bioconductor, rows are features and columns are samples. It has a familiar base API by the R software that can be extended by packages such as matrix stats. Matrices support matrix algebra and are compatible with BLAS and LAPACK, and they are C and C++ accessible, so you can write algorithms that work in these languages when necessary. And overall, they are quite conducive to interactive data analysis. But data are getting too big for ordinary R arrays. As an example, there is a single-cell dataset published by the 10x Genomics company that contains 1.3 million samples, or cells. It's one matrix with some 30,000 rows, or genes, and some 1.3 million samples. And if this was stored in R as an ordinary array, it would require more than 140 gigabytes of RAM. An example from my own work is analysing DNA methylation data from the GTEx samples, where the data would occupy more than 500 gigabytes in memory if stored as ordinary arrays. This is where DelayedArray comes to the rescue. For the 10x brain data dataset, this can be stored in memory in under 200 megabytes of RAM. Similarly, the GTEx DNA methylation data is just over 200 megabytes in memory. And how this is done is by keeping the assay data on disk. In these examples, in an HDF5 file that is wrapped in a delayed array, the focus of today's workshop. A delayed array still looks and feels like an ordinary R array. It inherits many of these properties, so it is structured, it has a familiar API, we can build packages that extend upon it, we can use things like matrix algebra, we can interact with these data sets from C or C++ using the beachmat package in this case, and it is even more conducive to interactive data analysis. So a delayed array refers to both a class, a package, and an extensible framework, and we will learn about all aspects of this in today's workshop. As I mentioned, it is part of Bioconductor and developed by Hervé Pages, and it is developed using the S4 object-oriented system, like most of Bioconductor. To install it, you need to install the BioC Manager package from CRAN, and then use it to install DelayedArray. The DelayedArray ecosystem is the 50 or so packages that depend on DelayedArray. These can be broadly broken down into user-focused or user-facing packages, developer-focused packages, and packages that users and developers should probably know about. So starting with the last one, the DelayedArray package implements the DelayedArray framework. The HDF5Array package extends that by allowing you to work with data stored on disk in HDF5 files using the DelayedArray framework. The delayed matrix stats package implements several useful row and column summarization functions of delayed arrays. The TileDB package, which is not yet available through Bioconductor, provides another on-disk backend for working with large data sets that is compatible with DelayedArray. The BioC singular package allows you to perform singular value decompositions or principal components analysis of these delayed arrays. The VCF array and GDS array have a different focus, looking to store genotype data on disk and accessible via the DelayedArray framework, whilst the RHDF5 client and RESTful SE packages focus on using remote data stores, such as an HDF5 file stored on a server, and interacting with it via the DelayedArray framework. Some common Bioconductor packages that make use of the DelayedArray framework include the Droplet Utils package for working with 10x genomic single-cell data, the Loom Experiment package for working with another form of single-cell gene expression data, the Scuttle package, which implements low-level utilities for processing single-cell data, the Bachelor package, which implements batch correction algorithms, particularly for single-cell data, and the BSSeq and MinFi packages for working with DNA methylation data. Finally, the Beachmat package is a more developer-focused package that allows a developer to write C++ code to work with arbitrary types of matrices. This introductory material is covered in greater detail in the workshop vignette, and I encourage you to read that in your own time. We'll now move on to doing some interactive coding to demonstrate the features of the DelayedArray framework. We will begin by loading the DelayedArray package into our R session. The DelayedArray package implements the core functionality of the DelayedArray framework. We will also load in the HDF5Array package. This package extends the DelayedArray framework to allow processing of HDF5 files. We will download an HDF5 file from the Experiment Hub. We load the Experiment Hub package and call the Experiment Hub function. we can then query the hub for the dataset we will use in today's workshop. Here, using the phrase 10x brain data. This returns a number of resources, of which we will use the highlighted one in today's workshop. We will download this resource and store the name of the resulting file in the variable fname. We can use functions from the RHDF5 package to inspect what is inside this HDF5 file. This tells us that there is a matrix with 27,998 rows and some 1.3 million columns that is called counts. We will now construct an HDF5 array, a type of delayed array, from this HDF5 file. We'll call this 10x and use the HDF5 array function to specify the file and the name of the dataset we wish to access. The data contain counts on nearly 28,000 genes and 1.3 million cells. This is roughly 100,000 times more samples than a typical bulk RNA-seq data set, and would require over 140 gigs of RAM to store in memory as a dense matrix. With so much data, we might expect it would feel sluggish to interact with this object, but that is not the case. For example, let's print the entire object to screen, something which is normally a very bad idea. As you can see, this has printed a preview of the data, as you might have experienced when using other Bioconductor objects such as genomic ranges, or other R objects such as data tables or tables. By now you might suspect that the 10x object is no ordinary matrix. In fact, it is an HDF5 matrix, which is a type of delayed array. We can see this by looking at the class of the object and by using the is function to confirm that it is indeed an HDF5 array. The data contained in an HDF5 matrix is actually stored on disk in the HDF5 file. Consequently, the 10x object takes up very little space in memory. We will now play around with computing on this data set. To make things slightly easier, we will first subset the data to the first 1,000 samples. Firstly, let's compute the library sizes for these data, which are the column sums. Secondly, suppose we want to know for each gene the proportion of cells with non-zero expression. We can do this using standard R commands. To compute the proportion of non-zero elements, we identify how many elements in each row are greater than 0 and divide by the number of columns in our data set. This gives us the proportion of non-zero counts for each gene. Finally, we can compute the median expression of each gene. Here we will quantify expression as counts per million. Here we normalize by the library sizes. Printing the counts per million to screen, again, we get the nice preview of the dataset. We can then compute the median expression of each gene using a function from the delayed matrix stats package. In particular, the median expression of each gene is given by the row medians of the counts per million matrix. These three examples highlight the power of the delayed array framework. Recall that the data in these examples actually live on disk in an HDF5 file, yet we interacted with the data and computed on it much as we would as if the data were in memory as an ordinary matrix. All three examples returned ordinary R vectors. The computations for these three examples made implicit use of the three fundamental concepts in the delayed array framework: delayed operations, block processing, and realization. We will now discuss each of these in some detail. We begin with delayed operations. Let's take a careful look at the 10x subset object. We can see that it is a delayed matrix rather than an HDF5 matrix. What has happened is that the subsetting operation that we performed has degraded the 10x subset object to a delayed matrix. The showTree function can help us see what has changed when we subsetted the data. If we look at the original data, we see that the tree of operations is simply the HDF5 data itself. However, if we compare it to the subsetted data, we see that it now contains a delayed subsetting operation. The subsetting operation has been registered in what is termed a delayed operation. Registering a delayed operation does not modify the underlying data. Instead, the operation is recorded and only performed when the delayed array object is realized. Delayed operations allow us to chain together multiple operations and only perform them as required. Here is a contrived example. Let's add 1 to every entry in the 10x subset. Let's look at the tree of delayed operations on this object using the showTree function. We see that in addition to the subset operation, we now have delayed this operation called a unary ISO op, which we'll explain in a moment. But it basically corresponds to adding 1 to every element of our dataset. We can continue our example by taking the logarithm of every element of our modified dataset. Again, we can print the tree of delayed operations. Notice that this has not added an additional operation. The delayed array package has combined the +1 and logarithm operations into a single operation. To continue this example, let's transpose the result. Again, we can view the set of delayed operations using the showTree function. Transposing the data has added this aperm delayed operation. Finally, let's realize a subset of the data as an ordinary array. Realizing the data will trigger the series of delayed operations for the subset of requested data. Here we realize the first 5 rows and first 10 columns in memory as an ordinary array. Many common operations can be registered as delayed operations. In the interest of time, we won't go into any greater detail, but the workshop vignette gives comprehensive information. We now turn to another fundamental concept of the delayed array framework, block processing. Block processing addresses the problem of if you have a large array and want to perform some operations on it, but can only load a subset of the data into memory. These operations could be element-wise or block-wise. As an example, suppose we wanted to compute the row sums of this matrix. One way of doing this would be to load each row of data into memory as a block, compute its sum, and then load the next row of data into memory and continue. Similarly, if we wanted to implement a col sums, we could load each column of data into memory, compute the sum, and then move to the next column. More sophisticated block processing strategies might load multiple columns of data into memory. For example, if we were wanting to compute the col sums, it may be more efficient to load multiple columns of data into memory and compute on those all at once, rather than constantly reading data from disk. We might also have an algorithm such as row sum that requires a variable number of columns. Conceptually, we could treat the entire matrix as a block. However, this rather defeats the purpose of using the delayed array because it will load the entire dataset into memory. There's also some notion of optimal blocks, and we'll return to these later in the workshop. These may be sub-matrices of our original matrix, perhaps corresponding to how the data are stored on disk. To better understand block processing, we'll start by using some functions that implement it to see how these work. To more clearly see block processing in action, we'll temporarily turn on verbose block processing. Let's now compute the column sums of the 10x subset object. Here I wrap the result in invisible so that it is not printed to the screen. With verbose block processing, we now see that the col sums is implemented using block processing. In this case, col sums has processed the data in 2 blocks, and the verbose block processing tells us that these blocks are over the rows of the 10x subset. Some of the most useful functions in the delayed array package implement common operations on a delayed matrix using block processing. For example, we can compute the row means of the subset. This is done using block processing. The delayed array package implements row sums, row means, row maxes, row mins, row ranges, and their column-wise equivalents. The delayed array package also implements 2 lesser-known functions, row sum and col sum, to compute column and row sums of a delayed matrix based on a grouping variable. Matrix multiplication is also implemented using block processing. Another useful package to know about is the delayed matrix stats package that implements the matrix stats API for use with delayed matrix objects. It provides more than 70 functions that apply to rows and columns of delayed matrix objects. If you've been following along with the coding examples, you might like to try out some of these functions for yourself. As we've seen, many common functions for operating on delayed arrays are already implemented in the delayed array package. Extension packages like delayed matrix stats and BioC singular further expand the list of functions that can be applied to delayed array objects. Nevertheless, you may find a situation where you need to implement this yourself. That is, you need to implement a function that uses block processing. The documentation on this is a little sparse at the moment, but we will go through some simple examples in today's workshop.
Block processing requires 3 key steps. The first is to set up what is called an array grid over the delayed array to be processed. The array grid specifies the block structure that will be traversed when processing the delayed array. Some functions you may be interested in looking at are the default autogrid, row autogrid, and col autogrid functions from the delayed array package. These functions can make automatic grids for block processing. For finer-grained control of the array grid, you may need to use the regular array grid or arbitrary array grid functions, also in the delayed array package. After setting up the array grid, the second step is to iterate over the delayed array using the array grid. At each step, we read a block's worth of data into memory, either as a dense or sparse array, and compute some statistic on it. The block apply and block reduce functions can help perform the block processing. These functions can even incorporate parallelization via the BioC parallel package. Once you've computed your block level statistics, the final step is to combine the block level statistics to get your desired final result. The final step is generally up to you as a developer to implement. As an example, let's implement a basic version of the colsum functions, where we define each block to be a single column. This function will take in a single variable called x, which is a delayed array. The first step is to set up our array grid. To do this, we will use the col autogrid function. The col autogrid function automatically sets up column-wise grids over our delayed array. Here, each block in that grid contains 1 column worth of data, as specified by the ncol argument. The second step is to load the blocks of data into memory and compute the block level statistics. We will store our block level results in the block level stat variable and use the block apply function to compute the column sums of each single column of data we load into memory. So our first argument is our delayed array. The second argument is the function that we want to apply once our data are loaded into memory. So this colsums function is the ordinary base R version of colsums. And the final argument is the grid, the array grid that we set up in step 1. The third and final step is to combine the block level statistics. In this case, it's as simple as collecting all our block level stats and returning them as a vector. The block apply function returns a list where each element is the colsums for 1 block of data. So by unlisting the result, we turn that into a vector. If we load our function, we can now apply our function to the 10x subset data. This may take a while. We can now compare our result to that returned by the colsums function implemented in the delayed array package. Now it's your turn to try writing a basic function that uses block processing. Try modifying the basic colsums function to define each block as a group of 100 columns. Alternatively, you might try to implement a basic row sums functionality using the row autogrid function. See the workshop material for further suggestions. To modify our basic colsums function to define each block as a group of 100 columns is as simple as changing the ncol parameter to 100. If we now load that function, we can compute the result. You probably noticed that using a grid that included 100 columns was much faster than a grid that used a single column. We'll return to this shortly. To create a basic row sums function is as simple as modifying our existing function by changing the col autogrid to be a row autogrid, specifying that we want to operate over the rows of our delayed matrix and that we want to operate over 10 rows at a time. We also need to modify the function that we apply to each block to be the row sums function. To realize a delayed array object is to trigger the execution of the delayed operations carried by the object and return the results as an ordinary or sparse array. We can realize data in memory or to disk. To realize data in memory, we can call the as.array function on it. For example, let's realize our 10x subset data. The resulting object is an ordinary R array. In your explorations with the data, you may have noticed that the 10x data contains a lot of zero values. We might therefore opt to realize the data as a sparse matrix. We can do this using a sparse matrix from the matrix package. We can do this using a sparse matrix from the matrix package. In this case, the data are realized as a sparse matrix, specifically a DGC matrix. Realizing in memory realizes the entire object in memory, which could require too much RAM if the object is large. Therefore, a large delayed array object may preferably be realized to disk. Here we will demonstrate by realizing to an HDF5 file, but we could also realize to another on-disk backend, such as a TileDB array. Realizing to an HDF5 file As an example, we will realize the data to disk as an HDF5 file. The process of realization uses block processing, which avoids loading the entire dataset into memory. To more clearly see the block processing in action, we'll temporarily turn on verbose block processing. To realize the delayed array as a dense array in an HDF5 file, we will use the write HDF5Array function. Recall that realizing a delayed array triggers all the delayed operations on it. This can be seen if we compare the tree of delayed operations on the 10x subset dataset compared to the 10x subset HDF5 dataset. Notice that the 10x subset dataset contains a delayed subset operation, whereas the version that has been realized to disk does not. The way we have used the write HDF array function here creates the HDF5 file in a temporary directory. This can be controlled by the user, however. Please see the workshop material for further details. Write HDF5 array also allows us to control how the data are written to disk, specifically how the data are chunked and compressed. These topics are discussed in further detail in the workshop vignette. Realization is an important topic with many subtleties. The workshop material covers some of these in greater detail. The main things to know about, however, is that realizing a delayed array triggers the execution of the accumulated delayed operations. If there are a lot of delayed operations, this can take some time. You can realize in memory as an ordinary array or a sparse matrix, or to disk, such as in an HDF5 file. Before we begin the Q&A session, I'll conclude with some general tips for delayed array-backed analyses. You cannot use the save RDS or save functions to save a copy or serialize your data when it is an HDF5Array-backed summarized experiment. Instead, you should use Save HDF5 Summarized Experiment from the HDF5Array package. The reasons for this are explained in the workshop vignette. This is because an HDF5Array-backed summarized experiment contains files on disks that the Save RDS and Save functions do not know how to serialize. The Save HDF5 Summarized Experiment is specifically designed for this purpose. Similarly, to load the data back in, use the Load HDF5 Summarized Experiment. The Save HDF5 Summarized Experiment will create a directory that contains a .rds file and an HDF5 file. This directory can be moved around your computer or indeed shared with collaborators, who can then load it into their R session using the Load HDF5 Summarized Experiment function. Once you've saved your HDF5Array-backed summarized experiment using this function, you can proceed with your analysis as normal. Any changes you make to the summarized experiment can quickly be serialized using the quick resave version of this function. Block processing is at the heart of much of the delayed array framework. As a general rule, fewer and larger blocks generally means faster performance, but higher peak memory usage. Using more smaller blocks will generally result in slower performance, but with the benefit of lower peak memory usage. The default block size is 100 megabytes, which can be changed with the set auto block size function. Increasing the automatic block size is often amongst the easiest ways to speed up a delayed array-backed analysis. Data stored on disk, such as in an HDF5 file, are usually chunked into hypercubes or submatrices. Chunking refers to how the data are physically stored on disk. For example, data could be chunked on disk by column, much as an R array is ordered by column. The ideal chunking supports whatever common access patterns you need to make of your data. For example, if you know you only need to process your data by column, then column chunking should give you the optimal performance. Generally, you don't know what access patterns you might need, or you need a combination of both row and column access. In this case, the default option of using hypercubes for the chunking generally offers the best trade-off. A common confusion with delayed array is the difference between the block geometry and the chunk geometry. The difference is as follows. The block geometry dictates how the data are accessed, whereas the chunk geometry dictates how the data is stored. When these two geometries align closely, then performance using the delayed array framework will be at its best. When working with large data, parallelization is an attractive option for speeding up your analysis. However, parallelization performance depends heavily on the choice of delayed array backend you are using, such as an HDF5 file, and your computer's hardware. Some general rules of thumb are that parallelization is never as straightforward as you think and never provides as big an improvement as you hope. Generally, parallel writing to files is a bit of a no-go zone, whereas parallel reading from files is sometimes okay. When working with large data, it helps to break your workflow up into stages. At the end of each stage, save your outputs, for example using the Save HDF5 Summarized Experiment function. It really sucks to have to rerun everything just to make a plot, and this is good advice regardless of whether you're using the delayed array framework or not for processing large data. We've now spent nearly an hour learning about the delayed array framework. Here comes the kicker. If you don't need to use a delayed array, don't use one. That is, if you can load your data into memory as an ordinary array or a sparse matrix and still compute on it, then you're generally going to have a better time doing it that way. However, when this isn't an option, when your data are too large to be stored in memory, then the Delayed Array framework is a powerful set of packages to help you get your work done. With that, I'd like to thank Hervé, the developer of DelayedArray and the HDF5Array packages in particular, and to apologise to him for butchering his name throughout this presentation. I'd also like to thank Aaron Lund, who's developed many packages that support delayed array objects, and Mike Smith, who maintains the RHDF5 and RHDF5lib packages. Thank you for coming along to the workshop today, and I look forward to discussing it further with you in the Q&A session.
I've been looking at the chat. I haven't seen any particular questions, but if anyone would like to jump in with one, please do. What are the best practices for parallelization for HDF5 arrays? For example, reading from a file within a BPL apply loop. So my answer is, honestly, I'm not entirely sure. So I've certainly attempted reading from HDF5 files in parallel, and in some cases it's worked, in other times it hasn't. And I think as a general rule, I would say it's probably not a good idea, but sometimes it seems to work. And the technical reasons for that, I don't fully understand. And I think it actually depends on how your HDF5 software has been installed and the particulars of the computing system you're working on. So I know that's not a very satisfying answer, and I hope that we can develop some more clear guidance around that, within the Bioconductor project. Can users specify the maximum memory usage, or should it be controlled by the chunk size of HDF5 in advance? So this is where the distinction between the block size and the chunk size is important. So the block size dictates how data are accessed, how data are read, whereas the chunk size dictates how data are stored on disk. So basically the chunk size will not influence how much memory is used, controlled by the block, which is how much data can be read into memory at one time. So as a user, you can control that by using the set auto block size function or lower level utilities. So you can specify a maximum, but it's important to know that that is just controlling how much data is read into memory. It doesn't take into account what is required to then compute on that data. For example, if you load in a large matrix and then you want to perform say a principal component analysis of that matrix, that memory required to do the PCA is not factored into the block size. And so you generally don't want to load in, and you don't want to have your block size to be the size of RAM. You need to allow some overhead for doing actual computations on the data. Curious about huge data sets. Any limitations you can think about around, say, biobank scale data, which is, say, tens of terabytes? Any thoughts about using performance in cluster environments, assuming performance scales both with the flavor of hard disk and network overhead if using NFS or similar? So the largest data sets I've worked on have it's from around about 200 patients, and in particular looking at non-CPG methylation. So that is a set of matrices with about 200 million rows and columns for the sample. And if that was in memory, that would be sort of more than half a terabyte. But on disk, particularly once compressed, it's far less. So I'm certainly not got any experience working at sort of terabyte size data on disk. My experience is that performance varies across different computing systems, and that includes how the file system is set up. My experience working on this was largely when I was a postdoc with Casper Hansen at Johns Hopkins, and there the cluster is very well managed, and I had a lot of support there, but I still struggled at times to fully understand why certain operations were slower than others. So that's partly the computing environment. It's partly format and library. As I mentioned briefly in the workshop, there's a new sort of kid on the block for disk-back array data called you may have seen a little bit about it. Dirk Edelbutel. Sorry, I'm butchering everyone's names today, but Dirk has been involved with the TileDB RRA package, which was briefly on in the whims of Cran and disappeared. But during that time, or overlapping time, Aaron Lund has developed a TileDB array package, which is on GitHub currently, that allows you to use a TileDB disk backend. So it'll be interesting to see how the performance of that compares to using HDF5 files, particularly for larger data sets and also sparse data sets, which to my understanding have a sort of native format in TileDB that's not present in HDF5.
Hey, Pete.
Yep, sorry, Martin, yep.
I just wanted to mention for parallel processing, maybe the model is like you have a reader, a function that reads from the delayed array and then hands off the compute task to a parallel backend. And that's implemented in, I think it's in the genomic files package. There's a function called reduce by yield. And you provide a yield function which produces a block of a realized matrix probably, and then passes that off to a function that does the computation and presumably.
Yeah, that sounds very promising. I think one of the challenges, so I've been using DelayedArray for probably 4 years now, so basically since it became part of Bioconductor. And the big challenge, like Hervé has done an incredible job, I think, with this, but it is certainly early days, it was difficult to reason about some of how to design algorithms for it. And I think as I've gained more experience and other people have gained more experience, you start to recognize some of these abstractions, like having a reader that passes off data to other workers that do the data, sort of the data processing, and trying to figure out what are useful and repeatable sort of abstractions will be of benefit to the project.
Give a shout out to the discussion of TileDB and the developer forum. And first of all, there's a channel in Slack for the developer forum, which has lots of interesting topics. And then the specific conversation about TileDB, I was going to say was in the course materials, but actually I think we failed to record that conversation. So I'll stop my plug. But the developer forum is a great place to learn about things.
I will go back and watch that. Unfortunately, being here in Australia, it's nearly 11 o'clock at night here, so I actually haven't seen much of the conference this year so far. I'm playing one video, so I look forward to going back. What do I mean about realizing a delayed array? So I went through that rather quickly in the spoken sort of presentation. There's a bit more detail in the vignette, but basically it means realizing a array means sort of trigger delayed operations and computing the actual result. So for example, when you do a subsetting operation or when we take the logarithm of a delay, it basically doesn't do any computation. It just sort of records the idea of taking a logarithm or doing a subsetting. And then when you realize the data, it actually goes through and triggers all those operations. So sort of by keeping these operations, there's opportunities for delayed array to simplify them. So for example, if you have a matrix and you take a transpose of a matrix and then you take the transpose. So for example, delayed array can recognize that and realize that that is an operation that doesn't need to be done at all and sort of essentially cancel that operation out. So realizing it is the triggering of the operations and into memory as an array or a sparse matrix, which means you can actually construct the result in memory as an ordinary R array, or you can realize the result to disk, which is useful when you have a very large data set because it can make use of block processing so that the whole data set doesn't need to be loaded into memory all at once in order to do the computations. So for example, if you had a large array on disk that you wanted to compute say the logarithm of, as a simple example, it would do the data set at a time, compute the logarithm, and then write that result to disk, and then read the next part in, compute the logarithm, write that to disk. If I haven't addressed your question directly, I don't know if these poll results are permanent or not. So if you perhaps want to take your question to the Slack channel, there's I think there's a delayed array channel on Slack. If not, there's a big data rep channel, which is probably the best place because you won't get just my sort of experiences of using delayed array, but you can hear from Hervé and Aaron and others who are essential to this work. So you can probably get even better answers by asking there. I won't look at that until I go to bed, sorry, until after I've gone to bed. So please post your questions there and I will try to answer them as best I can tomorrow morning my time. Thank you all for coming. Thank you for your questions.